Hello everybody, I am Dr. Farhan Zamir, an adjunct professor and academic specialist at Biotechnica, Bangalore. For today, we would try to understand a very important component with perspective of GATE. Now, at Biotechnica office, we get a lot of queries asking for, sir, how do we understand a very specific unit in the GATE syllabus and especially with bioinformatics. So, for this video, we will try to understand that very important component, how can we fetch more marks and how we can tactically answer the questions with regard to bioinformatics. Let's dive in. Hello everybody. So what we will try for today is, we'll try to understand what are the major tricks and tactics to understand and to attempt GATE Bioinformatics. And we will also see what are the very important topics to study in this unit. Let's start. Now, when I look into the GATE syllabus, the GATE syllabus says GATE BT. Now, GATE BT is GATE Biotechnology, and in GATE Biotechnology syllabus, we have 7 sections. Now, in this 7 sections, the 7th section talks about the recombinant DNA technology and the other tools in biotechnology. This is the caption of that particular section. Now, in that section, there are 4 major units, and the last unit is computational tools. So for today's entire video, we will try to see how best I can attend this bioinformatic component in the caption, that is computational tools, and how best I can answer my GATE questions. Before I actually go into the entire component of bioinformatics in perspective of GATE, I want to make you understand certain tricks and tactics. Okay, first of all, before I answer something, I first need to understand the question. Now, in most of the scenario, the GATE paper or any competitive examination paper is being set with a particular nomenclature or a particular set of rules, and this set of rules is called as Bloom's taxonomy, or it is also called as revised Bloom's taxonomy. Now, what exactly is this? Now, this is actually the schema for most of the competitive examination. Now, what do we do? So, Bloom's taxonomy is a pyramidal shape component. Let me try to make it much more clearer for your understanding. So, Bloom's taxonomy is a pyramidal shape pedagogy wherein we are trying to understand few important components. Now, there are 6 major levels in Bloom's taxonomy, and the first level, which we call it as L1 level, which will talk about the remembrance. So whenever I want to test the remembrance of a student, I am asking an L1 question. Or if I want to understand the understanding of a particular component, then I go on to L2. If I am trying to look into the apply component or that application component, okay, I would go on to the third L3 component, and analyze is the L4 component, wherein I would try to understand whether the student is able to differentiate between a given component. Okay, example, differentiate between FASTA sequence or GenPet format, something like this. And the L5 level is to evaluate. So whether the person or the candidate is able to evaluate the given question, and very importantly, L6 is the creative, the component. So here we look into the creative component of the student. So please remember, first of all, when I read a question, I should really understand whether the question is in L1, L2, L3, L4 or L5 or L6. So this becomes very, very important, because based on that, you need to fabricate your answer. Now, how do I understand this? Okay, sir, you said that you have L6 levels, but how do I understand which question is from what level? Now, for this understanding, okay, there are set of rules which we also call it as Bloom's action verbs. Now, Bloom's action verbs would talk about the various keywords which will define levels. So, example, if I am using a word as define, so define comes under L1. That is remembrance, wherein I am trying to check your knowledge. Now, if I'm looking into L2, I would ask a question which would start between, differentiate or estimate or explain. So this would try to, or this is, L2 question, which I'm trying to, look at your understanding component wherein you can comprehend. So this becomes very important. Then, if I'm asking an L3 question, please remember all L3 questions are application based. So, in bioinformatics, especially in this unit, people would try to understand what exactly, whether you are able to apply a component of biology and get an application out of a technology. So people would like to, or the examiner would like to look into this particular component, and most of your time, okay, when we look into the GATE question paper, it is either L3 or L4 questions. So here, the person or the examiner or the question paper setter would ask for demonstrate, analyze, divide, examine, all these kinds of action verbs. Now, the L4 is mainly to analyze, and analyze is, you're trying to analyze between two important components. Example, okay, arrange, break down, categorize, classify. This will make sure that have you understood the analysis part of the story. Now, the L5 question is based on the evaluation component. Now, in the evaluation component, you need to synthesize. People are looking into the synthesis of an answer, wherein do you understand a pipeline in bioinformatics? Those kind of questions will be asked. Example, judge or grade. All these kind of questions have been used, or these action verbs have been used. Now, the last and the most toughest level is the L6 level. And in this level, people would ask on compile, construct, create, design, okay, formulate, generate. All these kinds of action verbs are being used, and that is how you should know that the moment you read the question, you should know that, okay, I have been asked L1 or L2 or L3 level question, and with that question, what is expected with my answer? Okay, so this becomes very, very important. Now, with this understanding, I want to move on further. Okay, into the syllabus, and into the syllabus, when you look into it, as I already told you, it is the last unit of the entire syllabus. It comes under the section 7, and onto the section 7, it is again the smallest unit which has been mentioned. And for today, we will look into the entire syllabus of how exactly I can answer the GATE bioinformatics component. The syllabus starts with the caption as bioinformatics, resource and search tools. So here people are looking for your understanding on what exactly is database, what is bioinformatics, why should I use bioinformatics, what are the application of bioinformatics, and very importantly, are you able to understand the basic component which are of biological databases. Now, in biological databases, which are the major resource for bioinformatics data, there are major players. Okay, there are major databases which are called as the world's largest databases with a lot of biological information. Now, one such database is you need to have a thorough knowledge on NCBI. NCBI stands for National Center for Biotechnology. So this is the world's largest repository or deposits of biological database, okay, or biological information, which again, it is not just one database, but however, it contains an ocean of databases. It is an amalgamation of databases. So just make sure you go play around with NCBI. So once you understand NCBI, it becomes much more easier for the other databases to be understood. Then the next important database is your EMBL, that is the European Molecular Biology Laboratory, and then you have DDBJ, which is the DNA data bank of Japan. Then you have the structure database, which is PDB, that is protein data bank, and very, very important, you have SIB, wherein you have Swiss Institute of Bioinformatics, executing most of the proteomic kind of a database on XPCE. So this is the resource or the biological information which examiner would try to ask you and see what are the application part of it. Okay. Say for example, if I want to derive a literature, okay, and if I want to look into a particular, say for example, albumin. So I want to understand when was the albumin structure elucidated. So for this, somebody would have published it. Now, they would ask you this kind of a literature database, I would get it in DDBJ, PDB, or NCBI. So here, or PubMed. Okay, so here just make sure that even PubMed is a component of NCBI, but however, our right answer is actually PubMed. And PubMed refers, it is a literature database. PubMed refers to publication medline, wherein that information on when exactly the publication was, published on, or a manuscript was published on albumin, it comes from under the literature database. If I remember properly, I think it should be somewhere around 1984. Okay. The next component in the syllabus is sequence and structure database. Now, when I talk about the sequence and structure database, here people are trying to understand whether apart from the biological databases, we are just going in deep, understanding whether you know how to retrieve sequences and how to retrieve structure. Very importantly, the genomic structure, and very, very important is your proteomic structures. So people would look for your understanding on GenBank, GenPept, UniProt, and PDB. And this comes from the structure databases and sequence databases. Again, remember, for sequence and structure database, still NCBI is for good help, but I would rather go on SIB, that is Swiss Institute of Bioinformatics, UniProt, and then Protein Data Bank. So these would be the major structure, these are the predominant structure databases. But apart from this, we still have a lot other databases which would talk upon the structure and the sequence of the biological information. Now, here comes the next unit, and the next unit talks about sequence analysis. Now, when we're talking about sequence analysis, here people would try to understand what are the various formats, whether the candidate is aware of various other formats. Now, here comes three major formats. Okay, I want you people to get this component as embossed into your mind so that you will never forget this. Remember, all sequences or any kind of a data, okay, irrespective of genomic data, proteomic data, every data which has to get deposited in a biological databases has to follow relational management system. Okay, so relational database management system has to be followed, and for the first time it was Edgar Codd, C-O-D-D. So it was Edgar Codd who actually explained the concept of relational databases. So this relational database management systems will, this comprises rules which are from 0 to 12. So I have the rules from 0 to 12, and these 13 rules, okay, because again in a binary system 0 is also of a prime importance, so it is from 0 to 12. So in total there are 13 rules. Now, any database which wants to deposit its biological data, or just apart from biological data also, any database should follow the Codd rules. Now, according to the Codd rules, rule number 3 or rule number 4 talks about any data has to be arranged in a form of rows and columns. So when I say rows and columns, okay, so this is one row and one column refers to one cell. So what is a cell? If you just remember your Excel sheet, and that one unit in that Excel sheet is called as one cell. So remember, any kind of a data, if it has to be deposited onto the database for its retrieval, for its deposition, or for the management of the data, it has to follow Codd rules. Now, this is not another exception, wherein even your protein sequence database should follow the Codd rules. And in this, I have taken up an example of albumin sequence. Now, with this as an illustration, I would try to show you three major formats wherein albumin could be explored. Okay, the three major formats are, the first format is called as GenPept format. The first format is called as the GenPept format. GenPept format refers to, Gen is GenBank, and Pept refers to peptide. Okay, so this GenPept format is a numbered format. It is again arranged in rows and columns. Okay, so, and you have both complete and incomplete catapult concepts also. So the next component is, the second format is called as the FASTA format. Please remember, most of the tools which process the data, okay, or most of the applications or most of the softwares, they're compatible with FASTA formats. FASTA refers to fast all. So, and how do you recognize a FASTA sequence? A FASTA sequence is always recognized by a greater symbol followed by its accession number And then it will talk about what is the name of the protein and what exactly is the organism from which this particular protein has been related. Now, if you compare, now here could be an L4 question, differentiate between the FASTA sequence or and GenPET sequence. So there it was numbered, and here I don't see numbers. This means to say it is continuous. There it was split of rows and columns. Again, here, you should not say that the FASTA sequence is not into rows and columns, but if you see, this is one row, okay, and one column. So this one row and one column indicates one cell. So this means to say even a FASTA sequence follows chord rules. So these are the major variations, major differences between a GenPET format and a FASTA sequence. Let us try to see the third format, which is called as graphical format. Now remember, until now, you were actually looking at a particular sequence. Now, I am not looking at a particular sequence, but I am looking at a pictorial representation. I am looking at a graphical image of the entire sequence. Now, this kind of a data, this pictorial representation becomes very, very important. Now, if you look into the structure, which is an albumin, okay, and here it says albumin is made up of 3 major conserved domains, and this structure, which has around 604 amino acids, okay, so this has been subdivided into 3 major subdomains, or we can call it as, a specific motif on a particular domain will define the activity function of a given protein. So, this gives you a pictorial representation of how exactly a sequences could be arranged. Again, to have a brush up of it, we said that there are 3 major formats. One is the GenPET format, the second one is the FASTA format, and the third one is the graphic format. Let's move on. Now, once you understand the sequences, the next component in the syllabus is scoring matrix. How do I score? Okay, so now at this point, we need to understand why do I require sequences. Now, sequences are required, to know the match, to know the similarity or dissimilarity. So for this, what you try to do is, you try to take up one sequence, which is the sequence of interest, which is called as the query sequence, and then on the entire wardrobe of the databases, you have multiple sequences, and then this sequence is being aligned to various other sequences, and there's the to and fro, sequence alignment, and based on this, I have the best match, or there is a similarity or dissimilarity between my query sequence and the subject sequence. So for this, I need to know how much is it similar and how much is it dissimilar, and for this, I require statistics. So in statistics, I use a word which is called as, or a component which is also called as the e-value. So e-value, if it is 0, that means to say the query sequence and the subject sequence have a perfect match. Now, as the e-value goes farther, okay, this means to say, okay, the sequences are going under dissimilarity. So this point we need to also understand, and this has been done by understanding the likelihood between the two sequences, the parsomy component of between the two sequences, and how much is the match between one nucleotide to another nucleotide, or one amino acid to another amino acid. This is called as the scoring matrix. Let's try to move on to the next component, wherein understanding alignments also becomes very, very important. Here, there are 2 major, guidelines. One is called as weighted, and the other one is called as unweighted, scoring matrix. So here, we actually use distance score of results, and with this distance score of result, I know how far is one sequence from another sequence, or how close is one sequence with another sequence. Depending on this, I get a scoring matrix, I get a score value, and this score value is called as distance tree of results. Now, once I understand the scoring matrix, the next component is to understand what is the phylogenetic interaction, what is the phylogenetic trace value. So this sequence, how best it matches with the other sequence, I can look into its phylogeny, and very importantly, I can use tools such as MEGA, I can use tools such as R, which gives me a much more comprehended values and pictorial representation. Another important component in the GATE syllabus is to understand the 4 major components. That is, one, to look into the genomic component. The second one is to look into the transcriptomic component. Then the next one is to understand the proteome, and very importantly the metabolome. So all these four components becomes very, very important, and the logic is still the same, okay, wherein the raw material is different, and with this raw material the end product is entirely different. But however, the pipeline many a times it is the same. So if you go and actually look into our previous video on how exactly the molecular dogma or the modern central dogma molecular biology works, we have a detailed video on it. So just make sure you also refer that particular video. Then, once I understand the entire component, the next point is, how do I predict a gene, or how do I understand the functionality of the particular gene? And for this, a beautiful database which is called as the conserved domain databases, then to understanding the domain and the motif component, to understand the orthology, homology and parology components, all these databases are being used, and this will give me a funnel-like filtration. And with this funnel-like filtration, using certain subsidiary programs such as R, I get all these beautiful pictures so that I will be able to predict the functionality of a particular gene. Now, the next important component is, once I have the sequence, because until now I looked into the primary sequence, and this primary sequence is linear. So once this is there, because no protein will stay in its primary structure because of its stability issues. So now what we'll do is, this protein will undergo folding. Now when this protein undergoes folding, okay, then I need to understand which amino acid is contributing for what component, what domain, or what motif, what is the structural importance of that particular amino acid. And for this, okay, I have taken up an example of albumin, human albumin, which has 604 amino acid, and in this 604 amino acid, I have used a tool for secondary structure prediction, and this is called as GAR4. Now, with GAR4, what I see is, when I have processed, I need to just take the FASTA sequence, paste it into the GAR, and then it will give me this particular information. It says, among 604 amino acid, around 271 amino acid is actually corresponding to alpha helix. So, in this sequence, wherever you see HHH, indicates alpha helix. Now, the next component is, wherever you see the E, okay, so you can see E over here, this says that those amino acids are contributing for the extended strand, and wherever you see the C, alphabet number C, okay, then these are actually contributing for the random coiling. So if you look into the entire profiling of albumin, it says around 44% of 44.5% to be precise of the protein is contributing from the alpha helix, around 41% has been contributing from the random helix, and then very importantly you have the extended strand which is contributing for 13%. Now the next component in the syllabus is to also understand the 2D structures, and how do I convert a ligand 2D structure or a chemical moiety 2D structure into a 3D structure. And for this we use a simple tool which is called as Corina 3D. Now with Corina 3D you are able to have an interactive session wherein this is a Java enabled system, so you draw a particular structure which is in its 2D and then say update, and then you have a 3D structure of it. Then you can download it either in .mol file or .pymol file or JMol file, or you can download it in .pdb format. Now once you have this structure, okay, how do you understand the 3D structure? So most of the molecular docking studies would require this as a 3D system. So hence, converting a normal smile into 3D structure becomes very, very important, and understanding this also is a primordial factor for your examination. Now, once you have understood the entire, you have understood the target, you have understood the ligand molecule, here comes the pathway. Now, for understanding metabolome, a fantastic pathway is your KEGG. Now, KEGG refers to Kayatu Encyclopedia for Genes and Genomes, and here it talks about the systems information, it talks about the health information, it talks about the genomic information, and very importantly, the chemical information which are necessary for the building blocks of a given organism. Be it bacteria, be it plants, or be it human beings. Okay, this gives you a holistic picture of the entire metabolic pathway in a given organism. Next, there is a small mention of metagenomics also, and here comes the metagenomic pipeline. If I have a bacterial system which has been isolated, a consortium of bacterium which have been isolated from soil, okay, or from a rhizosphere, as a starting material, how do I understand what are the various organisms present in a given system? And for that, metagenomic pipeline is a very important component for our GATE syllabus. Then the last component talks about metabolic engineering and systems biology. So metabolic engineering talks about the upstreaming and downstreaming, and how can I use bioinformatics as one of the aid which can facilitate the upstreaming and downstreaming process, especially in terms of metabolic engineering. So for this, there are 4 major steps. One is to build, to test, then you have to learn, then design. So you need to actually, thanks to AI, that is artificial intelligence, people are trying to build an holistic machines wherein you have less amount of people which are required for the technology, and the robotic arms will do the purpose. So here the entire system has to be learned in terms of metabolic engineering. Take an example of citric acid and try to do this, and this would be a great help for you for your understanding. The last component is, there is a bit of mathematics into bioinformatics, saying that how can I use mathematical modeling to understand biology? And with this, which is called as systems biology, there is a small mention of systems biology, which has both biology, technology and computational systems, so that these computational systems and technology coming together, and how can they actually contribute for biology? This becomes a very important component, and currently systems biology is in a large boom, so 1 or 2 questions from systems biology could be expected. Now this actually ends the entire syllabus. I hope this is actually easy if you work it on it. But any point of time, if you have any clarifications or doubts, just feel free to leave a comment, and also do not forget to subscribe. And there are a lot of students, both at Biotechnica and from our online YouTube viewers, who would request for various videos on various topics. We are very open to shoot videos for you of your interest, because at Biotechnica we feel your achievement is our success. Thank you very much.